everywhere where the blood goes. So these are rare though. Most bacteria do not cause disease, but of course some of them can be parasitic. Some bacteria are autotrophs. We're going to learn about a group of bacteria in a little bit called cyanobacteria. They perform photosynthesis. They have chlorophyll. The chlorophyll absorbs sunlight, and with the help of water and carbon dioxide, they are able to make their own glucose and feed themselves. And when it comes to bacteria, let's not forget they're really healthy to ecosystems. Here's a picture of a food web, but notice near the bottom are the decomposers, the bacteria and the fungi. Again, they're really important at adding nutrients back into the soil so plants can live healthier and ecosystems in general benefit whenever bacteria and fungi are present. So the word respiration refers to how do bacteria deal with oxygen. And there's a few options. The picture shows that some bacteria are obligate aerobic, and it means that they must live in oxygen. If you look in this picture here, we have a test tube with a loose fitting cap on the top. Well, if the cap is loose fitting, that means that oxygen can still get into the test tube. It's not airtight. And notice in the picture how all those little dots, those dots represent bacteria. They're all congregated, they're all gathered near the top of the cap, where oxygen is most abundant. These bacteria need oxygen. And so all the bacteria are growing near the top. They're near the loose fitting cap, so a little bit of oxygen can get in and these bacteria can survive. Bacteria that fit into this category where they must have oxygen are called obligate aerobic. Another category of bacteria will be just the exact opposite. These bacteria, notice how the picture has changed. There's still a loose fitting cap. Oxygen can get in, but notice how all the spots are at the bottom of the test tube. These bacteria are trying to get away from the oxygen. They're trying to avoid it, so they've drifted down to the bottom. If these bacteria tried to live near the top, there'd be too much oxygen there because of the loose fitting cap, and they'd all die. To some bacteria, oxygen is a poison. So these bacteria are trying to escape the poisonous oxygen, and they live near the bottom. These are called obligate anaerobic bacteria. And the other option is bacteria that can live with or without oxygen, and here they are. These are called facultative anaerobic. You can also call them facultative aerobic. The point being is that they can live with or without oxygen. Now notice the picture. They're mostly gathered, the bacteria are mostly gathered at the top near the loose fitting cap, but there are still some scattered near the bottom. That means they can live with or without, but because they're mostly gathered at the top, I hope you can predict and conclude that even though they can live without oxygen, they would prefer to have oxygen. Cells are more efficient in making energy when they have oxygen. But if they're ever in a bad situation and there's no oxygen around, they can still survive. So here's a little a problem here, and hopefully you can come up with the answer to this problem. It says the bacteria that causes the disease called tuberculosis. This bacteria lives in your lungs. If it lives in your lungs, you should be able to conclude what kind of bacteria is it. And think about that. Every few seconds you inhale a gulp of air. So your lungs are constantly filled with oxygen. So hopefully you're able to figure out that the bacteria that causes tuberculosis is an obligate aerobic bacteria. It lives in oxygen. What I want to look at is this little diagram of the kingdoms of life. There are 6 kingdoms of life. Focus your attention at the two bubbles on the bottom. There's a bubble labeled eubacteria, and there's a bubble labeled archaebacteria. We're going to talk about these 2 kingdoms of life. The other 4 we'll mention another day. The reason I want to talk about these 2 is because there's 2 kingdoms of life which are prokaryotic, which means they're bacteria. Let's look at the archaebacteria first. Kingdom Archaebacteria. Okay, these are the extreme bacteria. You might even just hear them called Kingdom Archaea. Take off the bacteria at the end and you have another name for them. Kingdom Archaea, Kingdom And there's a couple of categories that I want you to be familiar with. One category of archaebacteria are called methanogens. And what's so extreme about them? It's, again, we said they live in extreme environments. What's so extreme about them is they live in anaerobic environments. They live in places where oxygen cannot be found. That's pretty extreme. Most organisms need oxygen to survive. So the reason they're called methanogens, like their name implies, they produce methane. So you typically find these in intestines and in the digestional tract of animals. Humans, we have these bacteria living in our intestines. They produce methane as they live in our intestines. And every now and then the methane inside of our intestines builds up, and it gets pushed out in a funny sounding noise. And we call that a fart. So it's kind of funny that our farts, the gas that we fart out, the methane that we fart out, comes from these bacteria. That's kind of interesting. I hope you, and kind of humorous at the same way. A second group of archaebacteria are called halophiles. Like it says, halophiles are bacteria that thrive in extremely salty environments like the Salt Lake in Utah, the Great Salt Lake in Utah. Also in the Mediterranean you have the Dead Sea. So the Dead Sea and the Great Salt Lake, they are just way too salty for most forms of life. And so even though the Dead Sea is called the Dead Sea because there's no fish in it, there's no aquatic plants in it, what you do find is bacteria, halophiles. These things thrive in salty environments. They actually consume the sodium and salt for energy. And here's the third group of archae-extreme bacteria, the thermophiles. Like it says, these bacteria live in extreme heat. They can live up to 230 in water, about 230 degrees Fahrenheit. That extreme heat is way too hot for most forms of life. It gets hot in the deserts. You know, some of the hottest deserts on Earth might be 120, 130 degrees. That 120, 130 degrees, that's too hot for, that alone is too hot for most life. But for these bacteria, it's even 100 degrees on top of that, so 230 degrees. The picture shows a deep sea vent, and what we were really shocked. In the 1980s, we discovered these deep sea vents were giving off all this extreme gases and extreme temperatures down there, and we were shocked to find that there were living ecosystems down here. And we were able to figure out the reason you could have big ecosystems all the way down here in the bottom of the ocean, even though there's no sunlight for plants to do photosynthesis. There's bacteria that live down here. The bacteria can feed on some of the minerals coming out of the vent. Other things would eat the bacteria, other things would eat the things that eat the bacteria, other things would eat the things that eat the things that eat the bacteria, and so you had a food chain and ecosystems in places where we didn't know it was even possible. So these types of environments are fairly new to our understanding. This was in, I think, the early 1980s when these were discovered. So here's just a little flow diagram that kind of outlines the three major groups of archaebacteria, or archaea. If you want more time to study this, hit the pause button. So let's move on to kingdom Eubacteria. There are two kingdoms of bacteria, the archaea, or the archaebacteria, which we just mentioned, and now the kingdom called Eubacteria. And these are the bacteria that are common to most environments on the earth. Whenever you wash your hands, this is what you're washing off. You're washing off the Eubacteria that you may have gathered from touching doorknobs and handrails and cell phones. And one important test that we can do to help us identify Eubacteria is what is called a Gram stain. G is capitalized because the scientist who worked out this process, his last name happened to be Gram. And what this picture shows, what this picture shows is two kinds of outcomes to a Gram stain. Some bacteria are either Gram positive, and the left of the picture shows Gram positive bacteria, and other pictures, the picture on the right shows Gram negative bacteria. Look at the right side of this picture. the right side of the picture, if you count the layers on the outside, you have a bottom layer of gold, you have just above the gold layer, you have a second brown layer, and just above the second brown layer, you have a third green layer. And so they actually have these 3 outer layers of defense, which makes them harder to treat with antibiotics. So if you have a bacteria infection, the doctor is going to want to figure out, do you have a gram-negative bacteria infection? Because if you do, your course of treatment is going to be a little more difficult. So the other possible result would be a besides a gram-negative bacteria, would be a gram-positive bacteria. And if the gram-negative is harder to treat, then these must be easier to treat. And if you look at the picture on the left, you can kind of see why. This bacteria possesses 2 layers. Even though that brown layer in the middle is a little thicker, it's really that third green layer that the gram-positive bacteria do not possess, and the gram-negative bacteria do possess, that make these harder to treat. And so the treatment that you will have to go through if you have a bacteria infection will be different. If you have a gram-negative bacteria, it's a little harder to treat. If you have a gram-positive bacteria infection, it's easier to treat. So this is one of the first steps that a doctor might want to do if you are determined to have a bacteria infection. And what they do is they do a gram test, a gram stain, and after the test is over, after the stain is over, the bacteria cells will appear to be one of 2 colors underneath the microscope. And like the notes say, if they appear pink underneath the microscope, then the doctor knows, oh, that's a gram-negative bacteria. If they appear purple underneath the microscope, that's a gram-positive bacteria. And the reason the colors differ is because gram-positive and gram-negative bacteria, they have different chemicals in their cell wall. They have a chemical called peptidoglycan in their cell wall. And so based on the amount of peptidoglycan the bacteria have, they will look either purple or pink underneath the microscope. Now this picture just kind of shows the general outline of a gram stain. I'm not going to expect you to memorize the order of the chemicals. It's just the general idea is that they'll take the bacteria, they'll grow a sample of bacteria that might be getting you sick, and they'll stain the bacteria with these various chemicals. In picture A, they'll stain the bacteria with a chemical called crystal violet. Then they'll stain the bacteria with a chemical called iodine. Then they'll wash the bacteria with alcohol, and then they'll stain the bacteria with a chemical called safranin. And that's when they can then look at it underneath the microscope. And if it appears pink or purple, they're able to determine if it's gram-positive or gram-negative.